What's your name?
My name is Favor.
Can you describe your project in 2 sentences?
Hello everyone, my name is Favor, like I said. My research is looking at mitigating drought stress in potatoes.
Thanks everyone, thanks James. I'll start interviewing the students. All of them are PhD students at different universities. Most of them have got posters in the place where we had food. So please, after this session, we have some time to have some coffee, grab a coffee and go and talk to them. And we'll start with the first student next to me. So, what's your name?
My name is Favor.
Can you describe your project in 2 sentences?
Hello everyone, my name is Favor, like I said. My research is looking at mitigating drought stress in potatoes, and I'm using antitranspirants. Antitranspirants is a form of biostimulant that is put on the plant to block the stomata so that the plant can conserve water during drought. Thank you.
And you said you have been 2 or 3 years in your project yet? Or?
Yes, I'm in my final now.
Have you found anything interesting and exciting results yet?
Yes, in the first year we looked at some parameters, and one of the parameters that has been consistent, the result of this that we've seen that has been consistent is physiological disorder, looking at reduction in terms of necrosis, reduction in terms of rosetting, and also in jelly end rot using antitranspirants. The yield in the first year was not really consistent, but after that we got some result. But for now, what we could say is that the physiological disorder, we've seen so much result from that.
Very interesting. And how do you want the industry to take your research further? What do you expect from the industry to react to your findings, or how would you ideally, how would you see your results applied in the real world?
Yeah, we all know how stress affects potatoes, right from the feed even to storage and after storage. And heat and drought stress is causing about 50% of loss. If industry would take it up looking at antitranspirants to mitigate drought stress, I think is a way forward. It's an innovation that we all need to look at. In our crop group in Upper Adams University, it has been used on wheat, on rapeseed oil, and they've gotten so much result. And if we want to look at this on potatoes, I think it's a way forward.
Thank you. And the final question is, we have lots of people from different parts of the industry here. We have researchers, farmers, and agronomists, and all specializations. Who do you want to talk to after this session?
I want to talk with people who are doing, who are having work on storage disorders, and who are also, I know most of the people here are dealing with potatoes, but I want to get a really practical experience, like if I could meet people who are able to absorb somebody like me on a voluntary basis, to have a practical knowledge on the stakeholder, to have a stakeholder experience, it will be good.
Thank you very much.
Thank you.
What's your name?
Hello everyone, I'm CUPGRA Conference. So, can you hear me? I'm a PhD student from Cranfield University. Well, my research is to improve the efficacy of organomineral fertilizer in a potato agronomy while improving the soil health and reducing the greenhouse gas emissions.
Nice. And you just started 2 months ago, if I'm...
Yeah, that's why I started October.
I don't expect you have much results, but what are you expecting to find, or what do you want to find? If the situation was ideal, what are the perfect results for you?
Well, OMF has a key feature like slow releasing nature, so I'm looking how I can fit OMF as an alternative for the synthetic fertilizer in a potato agronomy. If I get a positive results, I hope it will be a promising alternative for the synthetic fertilizer.
And can you explain to us what is the OMF?
Well, OMF is a combination of mineral fertilizer plus organic waste that which release like nutrition in a slow rate compared to the synthetic fertilizer. When you apply synthetic fertilizer, that releases immediately, but with the OMF, it takes time and supplies a steady nutrition for the plants.
So we're talking about slow release fertilizers.
Slow release, yeah, that's right.
Nice. And how do you expect the industry to react to your research when you finish it?
I would say like this is one of the best options because the feedstock to produce a synthetic fertilizer is getting low. So definitely we have to look for the alternative options. So I think this is one of the alternatives for the industries to consider.
Yeah, I think you also considering the greenhouse gas emissions,
Yeah.
and the generic fertilizers have a vast release of greenhouse gas emissions, and I assume we're looking at these organo-mineral fertilizers have reduced greenhouse emissions, so this is also something you should be considering. And from the audience, who do you want to talk to?
Well, my research is mainly based on field trials, so I'm expecting to talk with the growers that I can get their real world problems and some insights from them that will be really useful for my research.
Brilliant. Yeah, if you want to talk about slow release fertilizers and plant nutrition, just talk to Kevin after this session. Thank you.
See you.
Hello, what's your name?
Hello, I'm Regan. Thanks. Regan Harding-Smith, and I'm a PhD student at Harvard Adams University.
Can you tell us about your research project?
So I'm looking at endophyte grass, which are a symbiotic fungi that live in different grass species, and I'm going to use them as a cover crop to suppress potato cyst nematodes and stubborn root nematodes.
You also started recently?
Yeah, in September, so a bit new.
What do you want to find in your results?
But of course I want to find different strains that will benefit growers the best, so that will be able to be put into rotations. So obviously I want to find results, so I'm going to try and screen a lot of different ones for the chemicals they produce to find out which ones have the right amount of chemical to best suppress these pests.
Nice. And how do you want the industry to react to your research?
So I hope they take it on board and use it efficiently, and I'm hoping that in future, if it works for these nematode species, it might work for other ones and in other crops, so take it further.
And this is very interesting because I assume it fits into the regenerative agriculture and farming practices that is a trend these days. So who do you want to talk to other than growers?
So I kind of want to talk to everyone really, just get the best idea about growing potatoes and how I can incorporate my research into your growing practices.
Yeah. Thanks very much.
Thank you.
Hello, and what's your name?
Hi everyone, my name is Ronald Njoro. I'm also based at Harvard University.
Can you tell us about your research project?
Yeah, so I am trying to use chitinous amendments to reduce potato cyst nematodes, particularly in between potato rotations, so when you're not growing the potatoes.
And very technical question, what's the source of your chitin amendments?
So at the moment, I've been using chitin amendments from shellfish, but we're also trying to explore other materials because chitin can be found in fungi such as the edible mushrooms that we eat, like soldier fly, and mealworms, and other materials. So we're trying to explore some of these materials that are already being produced by other industries and try to use them to manage potato cyst nematodes.
And you are in your second year, if I remember well?
Yeah, so I just started my third year. So
You start month your third year.
yeah, I've just started it, so I don't know where I am.
So,
yeah.
what interesting results have you found yet?
So, potato cyst nematodes, naturally, according to some literature, they decline at a pace of about 30% annually. So, from my work that I've done so far, we saw that we could enhance the decline of potato cyst nematodes by, we can enhance up to 50%, which is really interesting in just the space of 3 months. But we've also found out that, one of the mechanisms involved in, the suppression of nematodes using chitin is the production of these enzymes. And so these enzymes can actually suppress the hatching of the nematodes by up to 40% in just, which is really interesting. So we're trying to see how we can now, optimize this, this decline and see exactly what comes out of that.
Brilliant. And how do you expect the industry to take your research forward?
I am hoping that because we have different industries, I know that at the moment, especially like the shellfish industry, they are being charged to dispose of their waste. But if we can actually incorporate some of this material into the agriculture industry, then that would be really good because we can reduce rotations, especially for farmers, and enhance the decline. So it could be like a win-win solution for two industries.
Okay. And from the audience, who do you want to talk to?
Potato farmers, everyone really. In like last year, I wanted to kind of look at people who maybe had issues with potato cyst nematodes, but this time I'm open, like Regan says, to speaking with everyone. It's always good to get different perspectives. Maybe someone has been working with this material, or someone is interested in working with it in the future. It's always I'm always here to hear what you have to say about that.
Thanks very much. And if you want to talk about alternative solutions for PCM, talk to
I have to say it's not an alternative solution, but it's an addition to what you're already working on, basically. Thank you.
Thank you.
Yeah.
Hello, and what's your name?
Hi, my name is Celestina Nyango. I am PhD student at Adams University, working in collaboration with Ferra Science.
And what are you working on?
Yeah, so I'm working on detection, identification, and management of root nematodes of potatoes. So generally, okay, now I'll just concentrate on the first part, the detection. So I'm doing survey on potato growing areas to see which species of root knot are currently in the UK. Yeah.
And have you found anything interesting or exciting yet? Or did you just start your research?
Okay, I've just started my second year. So I was able to get some soil samples, which I have done molecular analysis on them. And so far I can say that we haven't gotten anything alarming from the soil samples that I had. So we have the ones that are currently present in the UK. Unfortunately, there's an outbreak that was presented 2 months ago, July. Yeah, that is the Melodia Chitudai that is present, but it has been monitored, and from the monitoring it shows that there has not been any more detection on the same.
And what sort of detection are you talking about? Because we've seen drone and multispectral detection that Ankush has presented. So can you disclose to us what sort of detection or technique you're using?
Okay, so generally it's just to do a survey on the soil from the potato growing areas, and then we will do extraction, nematode extraction, and then I do microscopy to see if at all there's any melodogyne in the soil, in the extract. And then I've been using Tuckman PCR to go up to species specific, and also I've tried the digital droplet PCR, which is a new technique that is also part of my objective, trying to validate it to see how specific it is and if it is able to detect even the smaller fragment of DNA in the soil.
And how do you want the industry to react to your research?
Okay, right now I know there's a lot of politics concerning that area because of the outbreak, but like human being, when you are sick, normally you don't get medication. We have to know what the problem is, right? Yeah, so it is good to know what is ailing your farm so that you don't just continue putting on management practices and maybe you are doing the wrong thing. So when you know the problem, you are able to approach it in a good way, and then also it will help you to overcome, like we will not be able to continue spreading this disease, these nematodes, especially when it comes to the seed growers, because all the seeds come from them. So if that place has some species that are alarming to the potato industry and it is not managed early enough, then it is going to spread across the whole nation.
Brilliant. And you mentioned seed growers. Who do you want us to talk to?
Okay, currently it is a national program, so I will really be happy to connect to agronomists, growers, and also if they are open enough, they can send to me their soil. I will analyze them for free, and also I can also come to their farms if they allow me to go and do the soil sampling there.
We might also send you some samples from NIAB.
I appreciate it. Thank you.
Thanks very much. What's your name?
So I'm Brandon, and I'm just into my second year of my PhD now. So I'm working on creating bruise-resistant Morris Piper potatoes. I can see already a few reactions to that quite positively. So I'll be using transcriptome sequencing to then identify potential candidate genes for CRISPR knockout, and then hopefully from that regenerate some microtubers into full lines that I can then look at the knock-on effects of these edits.
I assume we are not expecting any GE potatoes from you yet, but how far are you in your project? What have you found yet?
So up until now it's been predominantly colorimetric analysis to do with bruise phenotyping, but I'm just at the stage where I'm designing CRISPR constructs. So I've got 3 genes chosen out of potentially 4. The 4th one is yet to be confirmed. I'm not sure what I can share on those genes yet, specifics-wise, but the idea is that I need to make the constructs, and then hopefully transformations will begin early next year, which I believe will be taking place at NIAB, but I need to confirm details of that.
And how do you want the industry to react to your research? And before that, what do you think? How are you seeing the future of the politics behind GE?
I mean, for my work, I hope that it becomes a bit more easier or more accepted. There have been a few events that I've been to where it seems to be from market data that gene editing seems to be more accepted than GM. So hopefully that trend continues further. With regards to kind of the industry perspective, if there's one thing I took away from last year, it's that growers don't seem to be a fan of Morris Piper in particular. So I'm hoping I can somehow ease that burden a little bit, maybe give you a more positive outlook on growing Morris Piper.
And who do you want to talk to other than seed growers? Okay.
At the moment, I'm looking for to speak to growers more. The reason being is because I'm looking at 2 types of bruising, pressure and impact bruising. One area that seems to be a bit more difficult to try and simulate is pressure bruising. So if there are any growers who might be interested in collaborating on a pressure bruising trial, this is a longer term trial, but I would be glad to speak to any of those.
Thanks very much. And if you want to talk about anything related to bruising, find Brandon. Hello, and what's your name?
Hello, I'm Lauren Eddy. I'm based here in Cambridge at the Health Science
your project on?
I work on potato and its relationship with valvascular mycorrhizal fungi. So this is a fungus that lives in the soil and forms a relationship with most plant species, where the plants will give the fungus carbon resources in exchange for key nutrients like nitrogen and phosphate. So I work on that in potato, but another thing I'm doing sort of separately but related is working on new gene editing strategies in potato, in particular how we can use viruses to deliver gene editing machinery more efficiently. So yeah, two sides of my project.
Interesting. And you're wrapping up your PhD. What exciting results have you got you can share with us?
Yes, I think what's really interesting is that we've shown both in the lab and in the field that colonization of potato by vascular mycorrhizal fungi is really dependent on nutrient availability, so how much nitrogen and phosphate you apply to your potato. And to me, this really shows that our agricultural practices really do have an effect on this relationship. And while we don't really know kind of whether sort of not being colonized, being suppressed colonization in these sort of high nutrient, high fertilization environments is sort of a big problem for the crop. I think it shows that there's big potential room for improvement. Then in agriculture, we haven't bred for positive mycorrhizal traits in potato or any crop species. So there's a lot of breeding work to do, but also agronomic work that can be done to making sure that we're making most use of this potentially really important avenue of nutrient uptake in agriculture.
And I've always been fascinated about the application aspects of research in general. How do you see the application of your research into fields, into hundreds of hectares?
Yeah, as I say, I think the agronomic aspects of it is really complicated and there's definitely things to be done, but something I'm looking at in particular is the plant genetics that control the interaction with the fungus. It's very much controlled on the plant side, and so I'm trying to identify genes that would be potentially useful candidates for improving the amount of benefit that the plant gets from the fungus. So using the gene editing technologies that I'm working with to do that, and so hopefully a bit more of a longer trajectory for that, but hopefully, use some gene editing to get some useful traits.
Thanks. And who do you want to talk to?
I'm really interested to talk to anyone who's interested in regen ag stuff or soil health practices, because all these things really do affect mycorrhizal populations in the soil. So, and then also anyone who's interested in gene editing, doing gene editing, wants to know more.
Thank you. And if you want to talk about regen agriculture and fungal relationship with plants, just find Lauren after the session. Thank you. Hello, what's your name?
I'm John Owen. I'm a PhD student going into my third year, another one from Harvard University.
And what's your project on?
I'm looking at using behavioural cues to try and hide the potato crop from invading aphids. So I'm looking at developing what they call a push-pull system, which is a two-pronged approach. So you're trying to hide the crop that the aphids are trying to find, and also trying to divert them elsewhere where you can either control them more easily or they cause less harm.
And what have you found yet?
I've found several ways that don't work. I think several ways that don't work. I think the key word is yet. So I mean, it's part of the process. You've got to keep going. So I've ruled out, or partially ruled out, some things, and I've got some ideas going forward that need to try.
I remember last year you presented some work before the conference about spraying the plants with different colours.
Yes.
Is that something you are still developing or getting?
That's part for the moment. I'm basically investigating. There's been various attempts in various crops to try and mask the potato crop. I've been trying some of the ways to try and get a handle on aphid landing onto some of the background colours that people have been trying. And I've got some interesting results on that. I've got ideas going into the new year for a new approach.
Nice. And how do you want the industry to apply your research?
Well, that depends if I can come up with something that works. I have got some ideas that can combine several ideas, and I'm also bearing in mind in what I'm trying to do is that I develop something that's actually practical, something that will actually can work on a farm. So I've been speaking to some growers and some agronomists, running some of the ideas I've got past them to see if I'm thinking about doing this. Is this practical? And that's got some ideas going forward.
And who are the people you want to talk to today?
Well, I'm doing a lot of flight experiments in flight cages, but I'm really like to try and upscale some of the ideas I've got. So I'm interested in speaking to anyone who's game for a laugh, game to help me try some ideas out on a sort of larger plot scale or a field scale.
So basically, people who are happy to host some
trials. Yeah.
Trials. Okay. Yeah. Thank you. And if you want to talk about aphid control, probably,
yeah.
Find Joan after the session. Thank you. Hello, and what's your name?
Hi, I'm Annika. I'm a final year PhD student here in Cambridge.
And what's your project on?
So I'm working with plant parasitic nematodes, and in my PhD I'm really looking at the nematode side, and I want to understand more about how the nematode parasitizes the plant to then stop it from happening.
And you are wrapping up, so what have you found yet, and what's exciting in your research?
So we found the first two master regulators of parasitism in the nematode, and what they do is they regulate the production of a bunch of these tools that the nematode needs to infect the plant. And we can show if we only take one of them away, if we knock it out or down, we can actually reduce the number of nematodes that make it into the host by 80%. So this seems to be a very valuable target that we want to further explore for nematode control. And then also importantly, on the other side of the coin, we can see that these master regulators in the nematode are actually activated by the host itself. So there's some plant molecules that trigger nematode parasitism, and we'd really love to explore whether we can breed for plants that do not trigger nematode parasitism and can take that away as well.
And how do you see that fitting in the UK agricultural system for potatoes specifically, and also talking about gene editing of the nematodes themselves?
So yeah, what I described is really a signaling cascade from plant signal to nematode parasitism, and we can show that it's important, and now we'd like to transfer that into the field and target it on any level. So one big area we want to explore is plant breeding. We can show that these plant signals that trigger it all vary in natural populations, so it might be interesting to look at whether we can breed against it. We could also try to target perception by the nematode of these signals, or we could try to screen for some molecules that target these regulators inside the nematode, so a small molecule solution.
So you're not editing the nematodes, you're editing a plant that will express.
Yeah, so one way we could try to edit the nematode is so you can express something in the plant, double-stranded RNA, that gets taken up by the nematode and then would down-regulate the gene in the nematode, so that's an option. But otherwise, there are not many tools to edit the nematode, and it would be very difficult on the soil, so I'd be more focused on the other levels.
And how do you want the industry to take this further?
So as I just said, we'd love to explore plant breeding and drug screening for molecules that could target it in the nematode itself.
And who do you want to talk to today after the session?
So naturally, I would be interested to talk to anyone that has an input on this, but especially breeders or people that are involved in drugs breeding, and also very interested in talking to farmers to explore what could be a product that they would actually be interested in using on their farm.
Thank you. If you want to talk about this with Annika, just find her after the session. Thank you. Oh, and what's your name?
Hello everyone, my name is Edwin Torere. I'm from Upper Adams.
And what are you working on at the moment?
Essentially, I'm trying to improve the relationship between growers, retailers, and processors. So I'm trying to develop a tool where we can detect what's happening underground. I think there was an earlier question about can we detect using thermal imaging what's happening underground. So hopefully if I come up with something which can help me doing that, So it's not just about detecting water stress, but the impacts of water stress on potatoes.
And are you in your second or third?
I'm going to my third year.
Going to third year.
Yeah.
And what have you found yet in your 2 years of project?
I think what's interesting is that the simplicity, I'm trying to simplify science to what is usable to our farmers. So I've seen the potential of what thermal imaging can do so that our farmers can have informed decisions in knowing what's happening underground, especially on disorders which are related to water stress. So I think there's potential for using thermal imaging in this industry.
So it will be applied in fields, and you want collaboration with also to our manufacturers or develop, or what do you want to collaborate with in the industry?
Because essentially I want to then develop a machine learning tool. So I need to have robust data from different environments and different varieties and so forth. So farmers who are working different varieties willing to share some data which they've used before, if not trying to create new data which can be used in the future. So network of growers, McCain's, PepsiCo, individual farmers, growers, I'm willing to have a collaboration, especially I'm going into my final year of collecting data, so that would be great.
And does that collaboration mean that you want to go and do some imaging in the field, or?
Yes. So I've used semi-controlled environment at the moment. So food experiments or environment give me a validation of what have happened in the semi-controlled. Is it also applicable to open environment?
And finally, again, who do you want to talk to specifically?
Everyone in here. But I have a pre-meeting with Matt from McKenzie. I haven't met him yet, but if you're around in the room, I will post a session. I will post in the room. We can have a chat about it. But also, I think I want to have a chat with Mark for irrigation as well, a consultant in irrigation. So, yeah. But everyone who's willing to have to cover that gap between America and the UK potato production, let's have a chat that aspect.
Thanks very much. Hi, and what's your name?
Hello everyone, I'm Maria.
And what's your project on?
Okay, so in potatoes, aerial determinacy is defined as the plant's propensity to continue leaf production after flowering. And determinacy spectrum goes from very determinate to very indeterminate potato cultivars. So what we have seen is that indeterminate cultivars require less nitrogen and transfer it more slowly from the home to the tubers than determinate potato cultivars to achieve high yields under similar growing conditions. So my project aims to explore the mechanisms underpinning determinacy and nitrogen requirements in potato crops.
And how do you expect that to be applied in the industry? How do you expect that the farmers and agronomists and readers will use the data you are producing?
Well, I hope the information or knowledge generated from my research can be used as an additional decision supporting tool for nitrogen management in potato crops. And I also hope that the industry supports more research related to nitrogen management and selection of cultivars that show greater nitrogen use efficiency.
And who do you want to talk to after this session?
Well, I would like to engage with growers, agronomists, industry specialists who have worked with indeterminate and determinate potato cultivars to understand a little bit more of the growing patterns, the management differences, and quality outputs. And also, I would like to better understand if the industry is moving towards a preference for indeterminate cultivars, or whether maintaining the diversity in order to achieve yield and quality demand in the market.
And what are the main varieties cultivars you're working on?
Right now, I'm working with the reference group, which are innovator and royal, as examples of indeterminate and determinate.
Yeah, and Maria is also taking data from the reference group, which is funded by CUPGRA and done on our site on NIAB, at NIAB. So thanks very much.
Thank you.
Standing up is overrated.
Last but not least, hi, what's your name?
Hi, I'm Em. I'm part-time, so I'm in year 6 of 8 of my PhD in Oxford, and my project is all about looking at waterlogging and waterlogging tolerance in potato.
And what have you found yet?
I found some quite interesting shifts in tuber distribution, so a shift towards initiating lots of very small tubers that don't end up catching up. So kind of similar but quite different dynamic to what Rob talked about earlier. I've also found very disappointingly that if you don't maintain your temperature control units, they break. So yes, maintain your temperature control units and employ greenhouse stuff, guys.
And how do you want the industry to take your research forward?
Well, my research kind of has 2 possible avenues. So I'm a very biochemistry pipetting transparent liquids into other transparent liquids person at the moment. And what I'm doing is I'm looking at a particular family of enzymes that sense oxygen. And so we're looking at whether that low oxygen itself is causative of this kind of shift in phenotype that we're seeing, or whether it's, something completely different. And then that response that we're seeing has the 2 potential outlooks of, one, if we can isolate it and cause that same response without the stress of waterlogging, we can shift towards smaller tuber sizes if that's desirable for the market, but we can hopefully also do the opposite and develop potato cultivars that are less likely to show this extreme shrinkage when waterlogged.
And I assume you want to talk to breeders?
I want to talk to everyone in the tuber initiation crowd. Yeah, if you care about potato size, hit me up.
Yeah, find them after the session. Thanks very much. Can you give them a round of applause? We're going for a break.
Yeah, just say you've been a great audience so far. You'll be even greater if you get back here by 4:15, not 20:4. So if you start at 20:4, that doesn't mean arrive at 20:4. It means arrive at 4:15. Thanks very much.
And just to quickly mention that we have plenty of time to find the students next to their posters or in the posters area, talk to them, have a chat, grab a coffee and go and see their posters. Thank you.